Next up, we have Professor Dan Peer. He is the chairman of NeoVac and professor at Tel Aviv University. His lab was the first to demonstrate systemic, high-efficiency, cell-specific therapeutic genome editing in cancer. He will be sharing his insights on RNA delivery, challenges, and innovations in gene expression modulation. So, please, Professor.
So with your permission, I change a little bit the title, although I don't see it here. So something needs to, yes, now I see it here. Okay, so thank you for inviting me. And I'll start by saying that as scientists, we always have to think outside the box, or as I want to prefer to think, without a box. And I'll start by, I do have 2 hours, right? I'm going to be very fast, don't worry. How do we solve problems, really, as scientists? Let's imagine that now we have a serious problem, that the car has fallen into the river, and we need to think how we're taking the car out from the river. And this is exactly what happened about 12 years ago when I was visiting Aarhus, so in Denmark. And I put those slides when I took pictures into the web, and it became crazy. This is not Photoshop, so seriously. The problem of a car that fell into the river. What do we do as scientists? What do we do as human beings? We need to think how to get the car out of the water. And what is the trivial thinking process? We'll bring a bigger car. And this is exactly what happened. So you can see here the nice small car that fell into the river, and you can see this bigger car. But I promise you, these guys are not physicists. They did not calculate the momentum here. And what happened is this. So you see, it looks very promising, right? You take out, you lift the car, and see their faces, they're very happy. But something happens. Now, we don't have one problem in the water, but 2 problems. How do we solve this? It's a question to the audience. A bigger car, exactly. Tristan, you bring a bigger car, and of course what will happen is this. Okay, so people really do not think really toughly about things. But I want to actually to tell you that the future is already here. And from an RNA standpoint, already 30 years ago, Mr. Bean, here, he told us, he explained that as an RNA, he needed a better template, somebody to copy from. So this was taken from the Sidco Mr. Bean 30 years ago. So you see this invention of RNA as a template from DNA is not new. We also know 10 years ago, which is much before COVID started, that the mother of dragon, she already explained the differences between double-strand DNA and a single-strand RNA very effectively. It's not anti-parallel, but still demonstrate the concept. So now to take us very seriously into this therapeutic area approach, which is basically claiming that we are in a new era, and this area started many years ago, but the focus is actually ongoing, and we can do many things with RNA, much more than we can do with other proteins therapeutics. We can silence genes, we can activate or upregulate genes, we can edit them, and in the future, we might be even replacing them. And we're very lucky over the years to be in the right place, right time. And we had made some impact on each one of those buckets. So we're the first, my lab was the first to show we can deliver siRNAs to immune cells in a cell-specific manner. That was in 2007 and 2008. The first to show we can deliver modified messenger RNA in an animal in a cell-specific manner. The first to show we can do in vivo genome editing in cancer. And also, a few years ago, the first to show that we can do bacterial mRNA. And I'll talk a little bit about each of those, give you some examples. So we can do many, many things with that. The challenges in RNA therapeutics and vaccine, as we all know, is twofold. There is the RNA story, which as a non-RNA person, I will not talk about, but we have heard already some very important issues about RNA, and we will hear more about this today. And there is the delivery side of it. So we need an appropriate delivery system, and it needs to have all the known requirements, nothing new here. Evading, immune activation and clearance mechanism, efficient entrapment or encapsulation. Carrier must internalize, RNA must be released. Specificity is always true, and the winner of those things have been, over the past few years, lipid nanoparticles, which are around for quite a lot of time. And everything counts, everything, right? Even the water. And this is a serious problem. What water should you use when making LNPs? So what is the real answer? It really depends on what will be your application. So we are going into very tiny details. As the analytical people always tell us, this is very important, right? Because everything matters at the end. And we have already on the market 4 approved, FDA-approved, EMA-approved vehicles. Okay, so the Onpattro was the first, the siRNAs in August 2018, then the BioNTech, Pfizer, and the Moderna COVID vaccine, the RSV vaccines. And this end of the year, we will hear a few more new things that are happening in the beginning of 2026 as well, mostly in the editing space, but also in vaccines. So it seems that this is a vibrant area. And what do we do in my lab? We have about 30 people, and we have 2 groups. One that are delivery people, and the other side, which are more of biology people. The delivery people, we do everything from lipid synthesis into generation of targeting moieties, generation of monoclonal antibodies, protein engineering, etc. And over the years, we've tried to push the field of what we call active cellular targeting of RNA payloads into specific area. And it all started somewhere in 2008. So in 2008, we were working on understanding how fully charged lipids that are fully charged, have fully charged amines, how the immune system sends them. They are non-natural lipids, okay? So we don't have in nature lipids that are fully charged positively. We only have negative or Switzer ions. So how the immune system cope with this? And we have discovered that they agonize those fully charged amines, toll-like receptor 4, and probably also toll-like receptor 2. And we explain with toll-like receptor knockout mouse, basically that the signal transduction going downstream through NF-kappa B signaling, so you have cytokine induction, interferon responsive genes are going up, at the end, cytokine storm. This could explain why many people in early clinical trials with siRNA felt shivering, very, very similar things to viral infections, which is basically a cytokine storm. It could be very dangerous, but it could be also mild. This pushed the field of pH-sensitive ionizable lipids. And at that time, somewhere in 2010, we had looked carefully, very closely to the DLIM family that were synthesized originally with Marco Piccolini and Peter Coulis, and then basically were tested in multiple stages of a phase 0 trials in Alnylam Pharmaceuticals. And I got a very talented postdoc from UNC, Srinivas Ramishetty, that started to look at how really we look at cationic head groups, linkers, and tails. And we start creating different structures that overall synthesize much more than 50,000 lipids and characterize them. And you can do many things. So Srinivas is now in Neovac in Oxford, and Heinrich will tell us a little bit about this later. But basically, you can do many things with that. So we start characterization of those LNPs, and over the years, we have learned a lot of things. For example, we have learned and published how to streamline the process of screening libraries. And that became a very important screening tool that we published about almost 10 years later to look at the screening strategy. And many of the academic labs and, of course, industry are using those schemes because it makes a lot of sense. How you define what is toxicity, what is efficacy, is expression is the only thing you measure, or other things. This is another paper, and the only reason I actually put it, because you can see some structure, but the most important is the cover. So you can see a very nice cover that Sumo Gunanaidu basically designed those lipids and worked with us on some applications, and they're in the manuscript, so I'm not going to talk more about this, but this manuscript gives you a hint about the difference between trophism, cell trophism, and organ trophism, where we define those difficult questions because it depends on your payload. If you put an mRNA that is a luciferase-based, or an mCherry-based, or a Krelox system, so a Cre mRNA, you will show different things in your expression. And there is a discrepancy that we need to understand why it happens. So how we measure this? This is your colleague here, Tristan, in a new paper that will come soon, basically review the method that you can use for RNA and for LNPs, and there are lots of things, around us. And the most important thing is to also understand edicts that could happen. And it could be oxidation, it could be peroxides, it could be many other methods, and there is a new paper from Sanofi who suggests another mechanism. But these are devastating in the drug development field, because if eventually those small pieces will be oxidized or they will be reactive, they will first could be dangerous. The second, it could generate new entities for our RNA that will be very unpleasant. I'm quickly shifting gear to vaccines and why this is important. So during COVID, we're very much interested in developing our own strategy, and we use an RNB for spike area with human FC, a vaccine that we show with an mRNA that protects transgenic mice that have human ACE2 receptors on them. And this work was done with a former PhD student of mine, Uri Ilya, a set of manuscript, but we already had a COVID vaccine, so we didn't have to run away and do something else, but we could get very nice survival data, very nice antibodies, even protection when we do this challenge in 3,000 PFU of the COVID vaccine. Fast response for highly mutated pathogens. This is a semi-joke because we were asked by Nature Nanotechnology to write some perspective about this in early time of COVID during 2020, but it was published only in 2022. So what is the relevance for this? You can see, for example, that we are only looking at Omicron 1.1, but they're already in 2022 Omicron 1.7. So I think the evolving pathogen story was, semi-looked at, but at least we had the privilege of aligning strategies that could be used for tackling vaccines. But if we're seriously looking at the pandemics or the history of pandemics, we see that COVID is actually representing a very small fraction of threats over the years. And the largest one caused by a Black Death, the bubonic plague, mostly in Europe, that almost whacked half of the population in Europe, 200 million people passed away. But look at the year. Internet was not invented, and many other things. So basically, we couldn't see this in social media, but it was devastating. And so many painters over the years decided that they will paint those things. For example, and this is one example out of many that is presented in Paris in the Louvre, and you can see the vision of the painter. This was made 200 years later, after the plague, but still the title of this painting is The Plague in Rome, so you can see how people basically behaved. And there was a lot of gold rush for mRNA vaccine during COVID. Basically, most of them were against viral pathogens and cancer, but there was very limited data on the platform applicability against bacteria. And, you know, we are living in an area that antibiotic resistance is around us, and it will grow and grow and grow, and this necessitates alternative countermeasures. So in that sense, I had a questions to Kathy Carico, and that was somewhere in November 2021. And I asked her that, I told her that we are working on bacterial vaccines, on bacterial, mRNA vaccines. And is she positive, or what did she think about, you know, this as a potential success? And Kathy said, well, it's probably not going to work. And the reason is that bacterial vaccines, bacteria brings their own proteins. They don't use the host proteins. They are very sophisticated, even the bacteria itself. Sometimes it takes the immune system weeks to understand where you have a bacterial infection, or days to weeks, I would say. So we decided, and one of her concerns was how to make those mRNA immunogenic of the bacteria, proteins. And we decided that this is a challenge, and we want to accept this challenge. And you can see some of my team members, in this case, all of them are women, basically said it's, this should be the challenge, let's go after this. And so a little bit about this bacteria. So pestis, or Yersinia pestis, it's a gram-negative bacteria, and it's allegedly the etiological agent of plague. It's highly infectious and lethal. One bacteria can kill one person. So you definitely don't want to work in a regular lab with this. You have to have really important means, normally BL3 labs and above. Millions of deaths throughout history, several hundreds every year. So still people are dying from plague in some areas. Recently in the US, there was a pneumonic plague patient who were probably traveling, came to the hospital and died immediately after a few hours. It's also considered by the CDC, the Center for Disease Control, as a bioweapon, a T1 select agent. There is no approved vaccine to plague, and it can be treated with antibiotics, but still there are some strains that are antibiotic resistant. So there are two main performances of this disease. One is the bubonic one, or the Black Death, and the other one is pneumonic one, much more difficult. Coming back to the painters, Nicolas Poussin in the Louvre, he also had this very nice image of the plague and how much people, died. And this is his vision of the Black Death between 1330 to 1350. He lived hundreds of years later, but still this was his vision of what happened. So there are still outbreaks every year of a few hundred. The last big one was in Madagascar, it was in 2017, but there are still a few around us. And so at the end, we started looking at the bubonic plague. The mortality rates, if not treated immediately, is 50 to 60%. The team had two graduate students of mine, Ido Kohn and Aurelia, now working in different places, and collaborators from the IIBR listed here. And there is another disease which is much more difficult, which is the pneumonic one, which basically 100% mortality within days if not treated. It's airborne, very unpleasant disease. Very short incubation time. Again, one bacteria kills one person. And so how you define antigens, how you really take antigen, bacterial antigens, and how you define them. And this is from a recent paper we published together with a group in Ghent, looking at potential antigen discovery methods in bacterial vaccine development. It's true for any bacterial, but more specifically for mRNA. Okay, so these are the potential, I would say, strategies for antigen selection. But we have used, for the first one, for the bubonic plague, a very primitive way to select our antigens. We used our biological knowledge. So we started by selecting and designing a very classical mRNA design, looking at F1. F1 is basically a chaperone, also called CAF1. It's a chaperone related into the bacteria. And we started by designing a simple construct. The simple construct had a cap, 5' cap, has a signal peptide, the sequence of the F1, also called CAF1, and a poly-A tail, which was 90 nucleotides, 90. Then we improve it to 120. And at the end, basically, we created a formulation with one of the lipids. This is the formulation. I don't know if you can see. These are the particles. Looks quite nice. And we had a vision. We're going to do a prime 5 microgram vaccine. Then 2 weeks later, we isolate blood and we do a boost. We wait 2 weeks, we do another boost, also take blood, and then we do a challenge with Gestis. And when we did the first boost, the first prime and boost, we found, and we just waited, that we don't get antibodies. Not in the first bleeding, not in the second, not in the third. There are no neutralizing antibodies here, which means that first, Kathy was right. So you give a Nobel Prize to the right people. But the second is you do see cellular activity. This is interferon gamma. Okay? So there is a T cell activity, CTLs, cytotoxic T cell, but you don't get antibodies formed. So we decided there is no need to do a challenge because no protection. Nothing will happen. So what do? You go back to the construct and say, maybe our antigen is not immunogenic enough. So there are some tricks in the field. One of them is to use a GC content, high GC content, and we use exactly the same idea here. And we found that after the third bleed, so after second boost, we start seeing antibodies in a way that are neutralizing. But this is not good enough, because by the time, it takes a long time, people will die. You need to be very fast here. So we did a challenge experiment when we found that 50% of the mice die within a few days. This is not a good vaccine. So basically, we decided to optimize the sequence again, and we chose two strategies. One, there was a paper showing that if you take out the signal peptide, you will be more immunogenic in an mRNA with high GC contact. So we strike the signal peptide. Or when you add a human FC, it prolongs also the expression. And so we repeated those experiments, and this time we found very early on that we get, even in the first bleed, we get neutralizing antibody. The level, if we get above 3, you're protected. Okay? These are logarithmic scale. So we were very happy. Of course, these are a positive control with alum. I didn't mention it before. It's a recombinant F1 protein or CAF protein with alum. Nobody wants to inject this today. Anyhow, we got 100% protection. So you can see here 100% protection, okay, from a prime and a boost. So you don't need to wait actually for the third one. The problem is, and then you become greedy. You say, I don't want to wait for 2 additional doses. I want to just give a prime and see if I get antibodies. Now, everyone who studies immunology will tell you that this is not possible, because it takes time for the response to come, and to generate antibodies takes about 2 weeks. So I'm telling you, highlighting something that we not fully understand, but we are working with others on this. How come that after a single prime with mRNA, only with the mRNA vaccine, it's true to all mRNA, you can get good response and you can get protection. Not in all the sequence, in all the sequences, but in this case, for example, for human FC. Single dose, 1, and then you're protected. Now imagine, this is very rare, right? It's not a disease that everyone will have. But imagine that you go to hospital, you hospitalize, and you get infected by all the bacteria in the hospital that are antibiotic resistant. And you get to the hospital, and if you have a mixture of mRNAs that can protect you, you get a single dose at the day that you hospitalize. And in a few days, you are protected. Maybe not fully, but you are protected. That will change the landscape of treating people in hospitals. Because today, many of them coming with one disease, specifically people that are immune suppressed, cancer patients, younger people that have immune issues, and that's a big, big, big problem. So this is one example. Then we shift gear to the pneumonic one, which is even more dangerous. Here, we saw that it's actually more challenging. So you need a bivalent vaccine. You need 2 antigens that will work, at least 2. And also we had to test different mice strains, because different mice have different antibodies. There is IgG1, IgG2, and they're actually different mice. So C57 black, bulb C, and CD1 behave a little bit different because 2 of them are inbred and 1 of them is an outbred. But at the end, we found that we get protection, and I'm not going to tell you all the story. You can read it in Advanced Science. It was also covered a few months ago, 2 months ago. But you actually can get very nice data with this, and you can show that the dissemination of the bacteria is being completely cleared here. So look at the bacteria here, at the control in bulb C and CD1, and the same behavior go to the other one. You can actually get blocking of dissemination. It's an airway bacteria. So this is a story, ongoing story, and Neovac has a lot of interest in developing these bacterial mRNA vaccines. So hopefully next time we'll come up with something even more interesting, but we are working in this direction. Now, what can we else do with LNPs? So we can do some basic science. For example, so Shushmita was a postdoc in my lab a few years ago, and she came after reading a paper published in Cancer Research in 2011, looking at a genome-wide siRNA lethality screen that identify a gene called CCAP5. It's a cytoskeleton-associated protein 5, which basically means it's a replacement for Taxol. It works on the microtubule. So this gene is a regulator gene. It's an interesting one, and there have been some papers also showing that these mitotic genes associated with poor prognosis in non-small cell lung cancers. So Shushmita wanted to use this, and she tried to look at the biology behind this. I'm making a very long story short, but basically this was published. What we found is that by silencing this gene, you are destroying the division of cancer cells completely. One single gene, which is unacceptable in cancer because we know that in cancer it's a multi-disease gene. Maybe this is an essential gene. So this affects the cell cycle, the mitotic spindle. It's a multipolar spindle formation, so it's blocked, and they look like flowers. And you can do a very quick work and show that injecting it multiple times in 1 mg per kilogram body, you can block this. This is ovarian adenocarcinoma xenograph model, and you improve the survival dramatically and also the tumor volumes. So you can do lots of things. Same year, we also showed this in myeloma cells in vivo, and was cover of Advanced Science as well. And now I'm shifting gear basically by telling you you can also fine-tune compositions in a regular LNP that will direct you to different areas. And this is predominantly the work of people in Neovac and also Ricardo Rampado, a postdoc in my lab that is starting his lab in November this year in Padova. And basically what Ricardo and the team has done is to play a little bit with the formulation. So lipids are very important, but also the helper lipids are important. So if you play with the formulation, you can end up directing your LNPs after IV administration, not to the liver, but predominantly to other organs. For example, you can direct them to the spleen and you can direct them to the colon. And why this is important? Because then you can express proteins that are important during diseases in the colon. More specifically, an example is in IBD, inflammatory bowel disease, in a model of DSS colitis. And this is just expressing IL-10, which we love as a molecule. Not sure this is the best molecule to use, but actually you get therapeutic effect here, and it also seems to be safe. So playing with the composition, not only with your ionizable lipids, is also very important to direct different cells to different areas. You can also optimize your linkers. So each part of the lipid is important. You can optimize the linkers as well, and this is again a combined work of Somo and Ricardo. And basically, they started to generate different linkers. And we found that two of them, a reverse amide and an urea linker, when you inject them IV, they are accumulating in the lungs, a little bit the spleen, nothing in the liver, it's empty. Okay? And then you want to know exactly where, so you use an AI9 mice, basically an Ecrelox system where it's TD tomato expressed, and you can use flow cytometry and sort the cells and look exactly which cell type will take it. And we found that surprisingly, one is better than the other. There is a lipid 35 here that is uptaken very nicely in lung endothelial cells and also in epithelial cells, but less in dendritic or myeloid cells. Look at the scale here. There is also an issue of the pKa of the lipid. It's not absolute lipid, it's the formulation, because the assay is not very analytic yet. Anyhow, and so if it's become a little bit more pKa that is above 7, we know that they have some lung trophism for different reasons. I have 2 hours more, seriously. Okay, I'm moving. So we express a protein here. It's a bacterial toxin, Pseudomonas exotoxin A, and that's it. I just want to show you some nice work that we've done in collaboration with Roche Basel, reshaping the oligos with LNP. I'm just going to say 2 slides on this. We use LNAs because Roche acquired Santaris, a known company of doing microRNA. They had a lot of reagents, and basically they gave us the reagents, say whatever you want to do with this is good. So we decided to see if LNPs can lower the dose of of oligos. You know, oligos in the clinic, you're injecting 10 to 50 milligram per kilogram body, which is unacceptable. So we did an experiment, few experiments. We did the PCR, very analytic, where we show that 30 milligram per kilogram of anti-HIF1 alpha, this is LNA, so it's locked nucleic acid, is comparable to LNPs with 1 milligram per kilogram. Very effectively. So you don't need so much LNA if you entrap. And we show that in a model of colitis, you get remarkable work if you use TNF-alpha. So basically a locked nucleic acid against TNF-alpha. And you can see that many of the cytokines are being affected, IL-6, IL-1 beta, this is the TNF. And also quantitatively, we have also other things, including the colon length and histology and everything in this new paper here. But actually, there's lots of other things. I wanted to talk about targeted delivery, mostly with antibodies, because I think it's important, but also with other type. So chemical conjugation is super important, but it has some limitations. Antibodies are not alike. It's very hard to quantify this in a way that you are happy with this, and sometimes you get creatures like this that you get FC exposure, and these guys will be fed quickly with cells having FC receptors. So we develop a strategy that we generate an antibody that target the FC portion of any antibody from the same isoclass. And that became a very important tool to do in vivo drug discovery because it's also controlling the orientation. So we call this the asset, anchor secondary single chain enabling targeting moiety. Two former graduate students were behind us. We produce it in bacteria, we purify this, and this is a linker. So you put your LNPs with whatever payload you want, and you have your favorite antibody, and basically with this, you go almost everywhere for the immune system and some other target cells. And you know, sometimes it's hard to find those guys, so you can modify. And we call this a LAND, lipidated antibody nanoparticle delivery. Basically, you generate your anti-single chain with this, or you can do it with a Fab, or you can do it with DIA-body or mini-body. Basically, you need only 2 compounds, and that without changing anything, but you have to produce it individually. And at the end of the day, you have this targeting strategy. So I'm not going to go into a lot of details because I don't have time. I'm just going to tell you that we are going after all those things in collaborations and with other things, and some of them we have published, some of them not yet. But basically, you can go after cancer, for example, with an anti-PD-L1, and we silence genes, and we play with this. There is one gene, for example, hemoxygenase 1, that has 2 roles. It's basically sensitizing chemoresistance in cancer tumors and modulate tumor myeloid cells. And due to time limitation, I'll not go through this. So it probably is interesting strategy. And then I want to do a little bit about editing, which I think is important. So these guys were behind the first papers that we published. And over the years, we published more. Just to think about why cancer, it's still the second leading cause. And if you go after essential genes that really, if you knock them out completely, the cell will die. That will be excellent target gene in cancer. Of course, many other things can happen, and you need to target 110% of tumors, but there is no perfect solution in cancer. So just to give you a sense where we stand, you need your sequence and your protein, and we do it in a way, okay, that what we want to do, I'll show you a movie at the end, so it will be easy to understand. We are using this one. So a Cas mRNA with a single guide, and it took us from 2014 to 2020 to understand what we are doing in an analytical level as well, and how much the relationship between the guide and the mRNA, how much is important, what is the volume, what is the ratios, and how important are chemical modification. Because without this, until the mRNA has been translated in the cell, your single guide is degraded, and you will have zero data, nothing real. So we worked on this for a few years, and we, together with IDT, were able to create modified single guides RNA. And quickly, I don't have time for this. Quickly, I'm going to tell you that we could show that we get very efficient genome editing in vivo with NGS that are 80% editing in tumors in vivo in a cell-specific manner. And at the end was a really nice experience. I don't have time, so I'm just going on to the end of my story. I don't have time for this. Look how many slides I have here. Unbelievable. So in terms of vision, you can do many things, but the next wave is not only controlling, this, also controlling gene expression. The next generation is how to open and close genes when you want this. And this is a relatively early collaboration with Jim Collins at MIT that developed those toehold sequences that you potentially can open and close genes. And I think it's very important. Unfortunately, I don't have time to share with you about circular RNA, but we've done tremendous work, and we generated a company named Rebox Therapeutics, which was the first in the clinic that do circular RNA, single dose of RNA that really gives you a wonderful therapeutic benefit for a very long time. But this is for psoriasis. If you want to read, this is another paper of circular RNA that we published a year and a half ago. And just to the end, there are two bottlenecks. One is endosomal escape, that we believe we know everything. Probably we need to be very modest because we don't know many things. It's depending on many factors, including the cell type, the delivery system, different proteins are going up and down, and how to control this process. And another serious important for us is understanding how the immune system sense not only the RNA payloads, but predominantly also the lipids. Again, many of them are non-natural lipids. They can signal, and they can signal classical danger signals. And we don't really understand all the signal transduction, but we are going to really be hurt if we don't really understand this part in order to develop the next generation drugs. And just at the end, I think this is our next challenge, muscular dystrophies. And what we try to align here is how challenging is this field. Because at the end of the day, there are lots of nucleotides on the market already, but none of them really is potent enough to treat different types of dystrophins, of muscular dystrophy, and predominantly Duchenne one, but also the others suffer from these issues. So this is my team. I want to thank them all. I want to also thank our collaborator, our funding agency. I have an advanced ERC grant that is almost ending in 2 years. And of course, you can imagine who is this guy with the bald one. And just ending my story, this is something that the ERC from our previous ERC asked us to do. It's an outreach for the public. So it's a very naive illustration, but I hope you can listen.
Cancer is a leading cause of death worldwide. Cancer cells divide without proper control due to genetic mutations within them. Most traditional cancer treatments are insufficient and cause severe side effects because they harm not only the cancer cells, but healthy cells as well. To make cancer treatments more efficient, we at Payloads Lab have developed special lipid nanoparticles. The nanoparticles are able to recognize the mutated gene within the cancer cell and destroy it. Allow us to explain how they operate. We insert 2 RNA molecules into the lipid nanoparticle, a messenger RNA that has the instructions to build the CRISPR protein Cas9, which acts like a surgeon's scalpel, and a guide RNA designed to direct the Cas9 specifically to the target gene. On the outer surface of the lipid nanoparticle, we attach a special linker we developed called ASSET. The ASSET linker binds an antibody that recognizes a specific receptor on the cancer cells. After injecting the lipid nanoparticles into the patient's body, the antibody directs the lipid nanoparticles specifically to the cancer cells, sparing the healthy cells. The Cas9 mRNA is translated inside the cell to the Cas9 protein, and then assembles with the guide RNA. The Cas9 guide RNA complex enters the nucleus and locates the mutated gene within the cancer cell's DNA. Once recognized, the CRISPR scalpels cut the gene and permanently disrupt it. The treated cancer cells can no longer divide, no matter how much they try, and eventually die. Our highly effective and precise approach can revolutionize cancer therapy and improve the quality of life of cancer patients.
Very optimistic, but we have to be. Thank you. Questions?
Thank you, Professor Peer, for this insightful presentation. Let's open now the floor for questions from the audience.
That was very clear, no? No. Very brave.
You mentioned earlier that your LNPs can detect whether you put in luciferase or EGFP or whatever.
Not the LNP. The cells can detect.
The cells can read off what's the message in the mRNA.
Yeah.
Can that be true, or is there something else that's based on?
It's a complete artifact. What we're suggesting in the manuscript is that people draw conclusions based on the wrong assumption. So they use mRNA. The example that we do, we inject into mice that are healthy mice. We inject mRNA with luciferase. And we show that the entire liver is being highlighted, luciferase. Then you want to see in the cell-based level how much hepatocytes are taking up the LNPs, right? So how do you do that? You use a Krelok system. So you use those TD tomatoes mouse, and you have to select the right cells. Unfortunately, and this is the greatest artifact in our field, there is no positive selection for hepatocytes. There is only negative selection. So let's assume you do like everyone else, and you take the negative selection, because you positive everything else, you sort everything out, and you stand up, you are left with your negative selection hepatocytes. In the hepatocytes, the amount of expression of mCherry is about 7 to 8%. That's it. So where the other are going, either we don't know how to select for hepatocytes, which is probably true. They're not very unique markers on this. Or they're going predominantly to macrophages of the liver, Kupffer cells. Can this be dramatically changing therapeutics? Probably yes. Because you can look at drugs like inclisiran, which is an siRNA GalNAc, against PLK, against PSK9. So it's a remarkable drug. It reduces your level of LDL dramatically for about 6 months. How much is reaching the liver? When you repeat the experiment with GalNAc, it's the same, 8%. So either we don't know how to select, which is probably true, or 8% is enough to have this huge phenotypic change. I don't have the answer. I'm just saying we need to be very careful in our assumption. The amount is very low. Now, we know that this data is highly reproducible by the companies that generate those drugs. They even don't know. So either we don't know, maybe we are underestimating. Maybe, for example, the liver takes much more hepatocytes, and maybe we need to have a better selection markers because we are losing many other cells in this negative selection strategy. Or that tiny fraction is enough to make a big phenotypic change. I don't have an answer. I'm just telling you, you have to be very careful when you use different mRNA with different markers, because at the end of the day, these are markers. One of the reviewers of this manuscript suggested that our M-cherry kills the cell. Could be, but we stained for cell death. We didn't see it. But could be that many cells are dead, and we couldn't take them. So we repeated this with GFP. Could be also that it kills the cell, but we get the same numbers. And so I think my take-home message is just, be careful, because when we draw conclusions, sometimes we can kill projects that should not be killed. Just because we don't know how to select, or we don't know what payload to use, or we don't use at least 2 different methods, which I think is crucial. So don't count on one specific expression system to show that you're there, because they're very artificial. Thanks.
Thank you. Any more questions? No? Then thank you,
Thank you.
Professor. And you can... We can